Hi, so thank you. I'd love to introduce Spiderwort Technologies to the group. I'm Stephen Hanson, so I wear one of the business development hats here at Spiderwort. So we are a private VC-backed biomaterials company developing cellulose-based scaffolds for a variety of tissue engineering applications. Before I get into our spinal cord solution, I'd love to just give a brief overview of what our biomaterial technology actually entails. So it's based on a decellularization of plant material and retaining structures that are found in the plant kingdom and repurposing them as those extracellular matrices or scaffolds for tissue engineering applications. We have a lot of flexibility around what these materials can do when complete control over the biomechanical properties that allow this tissue integration and other unique properties that make the materials perfect for being biomaterials themselves. So again, here's that versatility of the materials that I was referring to. So the formulation, we can have everything from a solid block of material to a hydrogel, and we have control over the porosity and the structures of that material that once we're ready to implant them. Importantly, these are based on cellulose. So this is a non-immunogenic material that is biocompatible based on years of experience in vitro and in vivo that just supports tissue integration and blood vessel formation within the materials. Importantly, during the pandemic, we developed an ISO 5 clean room where we can actually produce GMP quality of these materials in-house. And this has allowed us to achieve some of this the biocompatibility, so that ISO 10993 testing, as well as get our first formulation of the material to be tested in the clinic. So what is Spiderwort doing with these biomaterials? So we have three main buckets that we are pursuing in the company. We have CelluBridge for spinal cord injury repair, CelluJuve, which we're developing for soft tissue augmentation, and then we have our third category of pipeline or other applications of the platform itself. Of course, given the audience that we have today, it's the CelluBridge for spinal cord injury repair, which I'll be focusing on. Now, this probably doesn't need to be repeated to the audience in the virtual room today, but suffice it to say that SCI remains a persistent unmet medical need where there are limited options available to patients. It is somewhat comforting to see the number of companies that are present here today working on a variety of different solutions to address this medical need. So Spiderwort's solution is the biomaterial implant that we refer to as CelluBridge. So this is a solid material consisting of a number of microchannels that go all the way through the material itself to connect one side of the lesion to the other. And importantly, because it's cellulose, we don't have cellulases in our bodies to break these down. So these are long-lasting materials that are going to persist for an extended period of time. And we think that that's actually important in this indication, and that we want these fragile nascent neuronal networks to be supported long-term and not have resorbable materials which are going to disappear after 3, 4, or 6 months. Additionally, we have all these microchannels that are stabilized using fibrin sealants that are known to be used in medical applications. So the first piece of data that I want to show you was our rat work that we completed in partnership with the University of Ottawa, where the original technology came from. So in this is a complete transection model where we sever the cord of the rats, implant our material, and then look for impacts both on locomotor function and histological analysis. We can observe locomotor recovery in as early as 10 weeks. So if we see the rats on the treadmill, this is after 10 weeks of that implantation of our material. So we can see clear evidence of that locomotor recovery, whereas control animals are dragging their hind legs and tails completely. Now, of course, rats aren't humans, and we know that there's many steps that go in that translation to get towards the clinic. One of the big important milestones for Spiderwort as a medical device was to achieve this biocompatibility, this ISO 10993 testing status. It is often just seen as a tick box or for a device where, yes, we've done our biocompatibility studies, but I don't think that actually gives credit to the amount of work and effort that had to go into this testing, specifically that brain implant testing model that we had to develop. I am happy to report that the SoluBridge is biocompatible, non-irritant, and has all of the green lights to proceed towards the clinic. One of the next challenges in the translation of these materials is that rat model was a complete transection model, but we know that that's not typical of what the injury mechanism that is seen in the clinic. So we performed a porcine contusion feasibility implant where we're injured pigs by contusion using a set of weights from defined heights to understand how post that contusion we can get the implant and insert it directly into the cord. So this was a non-survival study, but we were able to get the implantation into the cavities that existed. Next along our translation continuum was our non-human primate model. So this I'll mention up front. So the in-life portion of this testing is complete. We are still analyzing all of the data from it. But essentially, this is a hemisection model where half of the cord of a monkey spine has been removed and replaced with SoluBridge, and then having the monkeys monitored over a period of time for both kinematic scores and eventually histology and the like. So we have the clear first endpoint here was a safety endpoint. So yes, that we could implant this material and have it not be rejected in the monkeys over a long period of time. And that milestone was easily achieved. Additionally, we are looking for hints of efficacy and locomotor recovery in the monkeys. Now, this is a challenging model in which to look at that because this is as I mentioned, a hemisection where half of the spine, half of the cord remains intact. So there is a high ability for the monkeys to learn and to to compensate for that injury mechanism. That said, we are starting to see some signals that are there, but I'd be happy to discuss those in further detail at a later point as we get some of the publication materials and to be honest, complete the analysis with those studies. Now, all of this translation from that original rat data to the clinic has been guided by a clinical advisory board. So we have four people with whom we meet with regularly who've guided us on various aspects of experimental design on the preclinical work, but also as we look towards the clinic. This is in addition to other key opinion leaders with whom we've been consulting over specific aspects of different studies. And really, this leads us to where we are right now. So what is the next step in that clinical translation? That's our clinical evaluation. So we are looking at an early feasibility study. So yes, so as I mentioned, we are a medical device, so slightly different pathway than the pharmacological route that many of the other companies would be pursuing. So we have filed our investigational device exemption a couple of weeks ago. So that's June of this year. This is for an early feasibility pilot study. So this is just a single arm of 10 subjects with a relatively stringent inclusion criteria of acute complete thoracic. So we're looking within that 4 days post-injury for the AIS-A's in the thoracic level of injury. Endpoints here, obviously safety is number one. So our primary and secondary endpoints are both the short-term and long-term safety and monitoring of SAEs. But we do have a number of additional exploratory endpoints to see if there's a hint of efficacy. So we're looking at things here such as respiratory function, sensory and motor scores, and as well as neurological level of injury. These are all measured using INSCI exams, as well as a more subjective quality of life assessment that occurs. In all of these, we're just looking for a hint of what an efficacy signal might look like, and where we should be able to focus our efforts in subsequent pivotal studies with the material. Now, I did mention that we could be ready to initiate this clinical study as early as Q3 or Q4 of 2025. And that uncertainty over when is that we'll be clinic ready after the IDE comes back. But as a biotech company, as you may imagine, that we need to arrange the financing for that study. So Spiderwort is currently out raising its Series B round. So we last closed a Series A round in early 2022 at 13 million, where we raised $13 million. We're currently looking for $45 million to support the preclinical and clinical development across all of those three buckets that I described earlier on. So roughly 40% of that is designated for cellular bridge or spinal cord injury projects, the bulk of which is the clinical study development, both the pilot and getting into the pivotal study for cellular bridge. My second ask for this group, so beyond direct dollars, so for investments, we are interested in partnership and collaboration. So we are a medical device. We are a pure scaffold that we are using therapeutically in the spinal cord injury population. We intend to pursue that as long as, and get that into the clinic and make it as simple as possible. But ultimately, we know that a combination of a scaffold plus some other modality of therapy is likely going to provide that best option for the patients. So we're actively looking for companies developing stem cells, drugs, growth factors, therapeutic antibodies, and even other interventions such as electrical stimulation and physiotherapy to identify those with whom there's that synergistic effect between the scaffold and the drug. So I know that there's a number of these companies in the rooms. Many of them we've interacted with already, but we would love to start having some of those conversations, even if it's about getting some of that early preclinical data wrapped up, so that once cellular bridge is in the clinic or gets towards approval on its own, that we can start thinking about what that next step on a collaboration might look like. So with that, I'm happy to reach out and talk to folks after. And thank you for listening and learning a little bit about that introduction into Spiderwort Biotech.